So today I'm going to tell you about the exquisite materials that nature makes with genetic encoding. And then I'm going to tell you a little bit about the materials that we make, that we get nature to make for us. We trick nature into making new materials for us. First, I'm going to start with my journey here. It's not unlike a lot of the journeys we've heard earlier in SACNAS. That's me on the left as a kid with my grandpa feeding the orphan lambs. On the right, that's me on the bench a number of years ago. That path from kid to scientist was not only not easy, but it just wasn't obvious, okay? So I knew I loved to learn. I had in my mind I'd go to college, but I had no idea what is really college? What do you do? And most importantly, I had no idea how the love of learning actually equated into a profession that paid you money, that fed your family, okay? I knew what a carpenter did, I knew what a cashier did, I knew what a bank teller did, but I had no idea how learning led to an income. I'm telling you this because I think sometimes it's easy to look at people who are just a little bit further ahead than you and think, oh, it must have been easy for them, you know? They must not have had to work full-time as an undergrad. They probably came from a long line of people who had bachelor's degrees. They probably knew exactly what they wanted to be when they went to college. Absolutely not. And I think that when we look and we imagine that it's easy for one person, and it's hard for us, you know, undergrad's hard, grad school's hard, we think, well, maybe this means it's not meant to be. I probably chose the wrong field. And that's really ridiculous because there's bumps along everyone's path, okay? The important part is set your path and just keep charging. What I did know as a kid is I loved nature. Okay, so I did wind up at the University of Utah, woohoo, and I didn't know what I wanted to be. I actually started as pre-architecture. I like to draw, I like to draft and draw buildings. I ended up in anthropology. Because of a math class for pre-architecture, I ended up pre-med. And along the way, some business classes. And in pre-med, I took a lot of biology, and obviously I took a lot of chemistry, and that's when I fell in love with chemistry, okay? So I knew I loved research, experimental research. I knew I loved chemistry. I knew I loved nature, but how they fit together, I had no idea. I was proctoring an exam for an organic professor, Bill Epstein, and he said, so, you know, you gonna go to grad school? I said, oh yeah. What are you gonna study? I have no idea. And he did something so important for me. He said, you know, your love of nature and your love of chemistry are not mutually exclusive. And what he did that was so important was he gave my interests, my passion, a name, okay? He gave me an identity to look for. He said, your interests are called bio and organic chemistry, biomimetic, biomaterials, natural products chemistry. And that did something really important. Giving me that identity helped me figure out what grad schools to apply to, who I would work for at UCSB, and what I would do at this point. And so we'll get back to giving it an identity, giving your interest an identity. Let's talk about one of my passions. That's nature, okay? And nature really makes these exquisite materials through genetic encoding. On the left are the magnets from magnetotectic bacteria. In the middle, you have these silica frustrals from diatoms. This is amazing. This is an organism that literally lives in a glass house, okay? On the right, you have the backs of beetles. And those backs of beetles are not necessarily pigment color, but are actually structural color, okay? So what do these very diverse three examples have in common? And that is materials in nature are programmed, okay? Each one of these organisms has genetic information, the DNA, that tells that organism how to make that material. And those materials that are genetically encoded end up being very precise. They're made in minutes to hours. They are incredibly reproducible. And it produces these materials with not only defined structure, but defined property. And so our interest is not necessarily to mimic these types of materials, but to steal from nature a bit and to genetically encode different materials. These are called nanoclusters. I'll tell you about that first. And the other are genetically encoded polymers. First, a little recognition. I'd particularly like to acknowledge Reg Rocha and Jim Werner, two great colleagues of mine. And then the work was primarily done by Jazz Sharma and Tim Ye. All right, chemists, we learn that chemistry, we spend a lot of time of chemistry at the molecular scale. But in fact, if you look over this scale, there's chemistry all across this length scale. One of the things that we study in the Center for Integrated Nanotechnology is, of course, nanomaterials. One of those are nanoparticles. What's a nanoparticle? It's a collection of thousands of atoms of gold or silver, okay? And because of the size of this material, as well as the electronic communication between those atoms, this particle has a very rosy or purple color, okay? And in fact, it's been used by chemists and glassblowers for years, since the 4th century, to create pigment into different materials. So now imagine you take this particle that has thousands of atoms of gold or silver, and you start shedding atoms, okay? Now you shrink. You go from thousands of atoms down to just a few atoms of gold or silver. That's a nanocluster. So you're about 1 nanometer. You maybe have tens of atoms of gold or silver. What's a nanocluster? A nanocluster is a collection of a few atoms of gold, silver, platinum. It's very small, which is ideal because we want it to be smaller than the size of the protein or the DNA that we're going to use for detection. Because of that small size, it no longer has that rosy color that we saw in those big particles. Now it takes on very different properties. Now it's actually fluorescent if we make it right. And now we can make these clusters, not only because they're small, but because they're very bright, we can actually use them for biological sensing and biological imaging. So one of the things that we use to make these clusters is DNA. So we can genetically encode their synthesis. And the great part about DNA is that, of course, there are 4 bases, okay? And each of those bases is very amine-rich, and that's important for coordinating these metals. Of course, we can alter the order that we put each one of these bases, and that's the sequence. And what we found out quickly in making these clusters is that the nucleic acid sequence, the order of those bases, actually determines the cluster size. It's like the frame of your house, right? You build this frame, and you can only grow that cluster so big, okay? So we were able to see that the cluster size is determined based on our DNA template. We also saw that the nucleic acid determines the fluorescence of that cluster. Each one of those different colors you see is actually a different piece of DNA, and each one of those DNAs makes a very different cluster. We made these, they're genetically encoded. They're not only small, a nanometer or so in size, but they're highly fluorescent, upwards of 90% quantum yield. This is actually just an image a colleague of mine took where he tracked one cluster in time and space for quite a long time. All right, we've used DNA to make this cluster. But remember, DNA is kind of like Velcro. You've got the sticky and the fuzzy ends, and you know Velcro comes together. But DNA beats Velcro in infinite number of ways, but one of them is that DNA knows which way to bind the sticky and fluffy ends, right? So not only can we use DNA to encode this cluster, but we can actually use another piece of DNA as a handle, and that's a handle for detecting DNA for biothreat reduction or for tuberculosis or for influenza. This is a story of serendipity. I'm telling you this because there's a lot of serendipity in science. It's just about whether you're ready to see it and accept it and use it for something else. We actually had no intention to make this what we call a nanocluster beacon. We thought we knew very well how to make a cluster. It was early on. We accidentally made a dark cluster on a piece of DNA. That's on the lower left that you see. And that dark cluster was attached to another handle of DNA. And Tim Ye was very tenacious. Even though it was dark, he went ahead and stuck those 2 ends of DNA together. And a little piece of overhang, the G's, were around and actually turned that cluster from dark to a little bit bright. And what he noticed is that as he changed what we call that enhancer, he could change how those clusters communicate much better, and he could actually change the color from dark to very, very bright upon hybridization. This is the nature of what we call a nanocluster beacon. It consists of 2 parts. You have a dark nanocluster attached to the piece of DNA you're interested in. Let's say it's tuberculosis, okay? Also in solution, you have one of these magic enhancers, also attached to another piece of tuberculosis DNA. No TB in solution, they randomly rock, they don't talk to each other, it's dark. When TB's in solution, they've been designed such that they actually bind in close proximity, and now they turn on. It can be used for cancer diagnosis, for influenza diagnosis on surfaces, in solution. And in fact, by just changing this little enhancer piece, we can not only make one color of a cluster, but we can actually make a whole palette of clusters so that we can hopefully use those for multiplex detection. Not only does it detect a single bigger piece of DNA, but it actually detects single base changes, so single nucleotide polymorphisms, or SNPs. These are, of course, single nucleotide changes in our DNA that not only allow us to fingerprint and see the difference between you and me, but also to potentially indicate our susceptibility for disease. So these nanocluster beacons are able to detect these single nucleotide changes just because that single nucleotide change causes a shift in position between this enhancer and the cluster, okay? And that just really subtle shift changes the color that you see, and that's something we can see by eye. So I hope you can appreciate with this example that this is chemistry, materials science, and how it uses biology to impact medicine. Finally, I want to end very quickly with a story about genetically encoded polymers. I'd like to thank Mia Talilo, Kaushik Ghosh, Devin Close, and Eva Balog. All right, so in general, we need new methods to create materials that are going to go in our body. Okay, so our general goal is to create the methodology to create libraries of genetically encoded polymers that are important for regenerative medicine. So what do we envision? Like a lot of people, we would like to come up with a polymer that we could 3D print. We could harvest our fat, our stem cells and our fat, and then dump them in there, and it has all the biological, physical, and chemical cues to quickly allow us to make a new ACL. That'd be ideal. But here's the issue. Most of the polymers and most of the hard matter that we put in our body were never meant to go there. Acrylates, titania, stainless steel, those are made for airplanes and tables, right? So what we're trying to do is develop the methodology that allows nature to tell us which polymer it likes for a particular function. Without going into a lot of detail, there are a lot of advantages about genetic encoding of polymers. The first is that it allows us to mix and match and change that polymer very quickly. We can do it in our mind, we can type it in, we can order the DNA. It also allows us to make very large libraries of polymers. And those large libraries allow us to winnow down the polymer that has the functionality that we're looking for. We've used this technology to do a number of things. We've used this technology to find polymers that allow stem cells to quickly go to cartilage, and which we can start to spin cast and print and use for that ultimate application. We've also used it for a completely different purpose, and that's for optical materials. We can take the same sort of techniques and actually get nature to make different type of optical material. And these are optical materials that as you stretch and poke, they change their fluorescence. And we hope that those are going to be likewise usable for implantable sensors and electronics. All right, so I hope that you can see that genetic encoding is very powerful, and we can create materials that are well beyond what we have in nature. That genetic encoding allows us to make nanoclusters that are of defined size, that are defined fluorescence, and that allows us to have a handle to use those clusters for detection and imaging. And it allows us to make these polymers that are incredibly precise, that we can use for regenerative medicine, and we can make through a green synthesis. All right. Finally, I'd like to say that if you're like me and you didn't know what you want to be and you're still finding your path, that's okay. It's okay not to know. And you're going to eventually find it. You just keep going, okay? And the next is that it's a favor. So help me help the younger kids in our community put a name on their passion, okay? To identify, to put an identifier on what they like to do, what their passions are, and how that relates to STEM. I think if we can do that, we can get some little kid saying, Jen, I'm a physicist. Great. Now he watches a video on physics or checks out the right book. If we can start that early, I think we can get a lot more kids interested in STEM. Next, if the things I've talked about interest you and I can help you with your path, this is a very diverse field as far as STEM technologies, right? We need new instruments to be able to image the hard and the soft interaction. That means engineers, physicists, physical chemists. We need to be able to study and create new materials. That's inorganic chemists, that's organic chemists, material scientists. We need to define what the mechanisms are in these natural organisms. That's biochemistry, that's molecular biology. We need to discover new organisms that can do very unusual things. That's ecology, right? So there's a place for everyone and everyone's interest in this really interesting field. And with that, I'd like to say thank you, SACNAS 2015, although I put 2016. Thanks.